So we'll start with good and bad news. And the only bad news is we're getting closer and closer to the end of this wonderful event. Some consider that even good news because we're all exhausted. But you're still all here because the highlight of this very ESG 2025 will happen now in the next session. So my name is Alex Heuschen. I've been sitting on this podium for way too long already. You know me by now. But it's a great honor to co-chair this session with our new president of the ESG, and she will introduce herself.
Martina Cornell from Amsterdam, and I'm indeed the president of the European Society of Human Genetics since yesterday. So it's an honor to chair this symposium with Alex.
And as you all know, we have the great honor to give prizes every year, and it's 3. And one exceptional laureate prize lecture you've already seen on day 1 as a first highlight, and now we follow with 2 more. And those are the Mendel Award and the ESG Award. And this year, we're extremely privileged to have greatnesses here on stage. Maybe one person you know extremely well because he developed a tool that we all work with, and we'll come to that. But we'll start today with Katalin Karikó. Before I say anything more about it, it's a great honor and joy that my good dear friend Alex and co-chair of the ESG, Alex Raymond, will actually speak some words of laudatio. The floor is yours, Alex.
Obviously, Alex wants to stay on and on and on at this conference. Thanks, Alex. So, I'm Alex Raymond. I'm co-chairing with Alex Heuschen. And first, many thanks for all of you to have come here to Milan. Since 2006, the European Human Genetics Conference closes with a lecture of a distinguished speaker. And 3 years later, it was officially named Mendel Lecture in reference to Gregor Mendel's research. And today, we are absolutely extremely honored to host a true trailblazer, and I will come back to this. And this is Katalin Karikó, Kati. And Scandinavian would have said her life is a saga. Gli italiani e I greci diranno è un'odissea. I think she should say, and I will do my best, Ka landos elete rege hinbe hillo. Her adventurous life is worthy of a novel. And why so? Because she studied in Szeged. Oh, sorry. She studied in Szeged. She crossed the Atlantic as a young mom, then went through, maybe she will say a few words, maybe sometimes some tumultuous years from Temple University to Bethesda Naval Hospital, then to University of Pennsylvania, and then basically joining BioNTech. And so she was stubborn enough to do not going on when papers were rejected, when grants were rejected. So please take note, be stubborn like her. Because rejection might come back, but be, strong and continue. And I have to say, because we are in human genetics, that this is a trait that is going from different members of family, because her daughter, she just said to us at lunch, early on, she said, I will be in the Hall of Fame of my high school. And basically she did, and she did it multiple times, as she is also multiple time Olympic medalist. So genetics has some interesting features here. And so maybe I should finish with this. You all know the head of the story. You have some legacy in all of you, injected in all of you, in 2020, 2021, or 2022. We are extremely honored to have you.
Thank you. Thank you very much for this nice introduction. Just to prove that I get the Nobel Prize, I get this with me here, just to show you, before you get your own. I just used to carry in my pocket, you know, like security guy let me in today without the badge. Yeah. Yeah. So I am so happy to be here, and I have to say I never talked to that many scientists. So, fellow scientists, I am very happy to be here and talk about developing mRNA for therapy. I was thinking, how should I start with my life or something, but because uncertainty in science dictated me that I have to say something about, you know, we do discoveries in academia, in research lab, and then maybe we form a company and doing more testing, and when we need more money, then we go to, you know, the big companies and run clinical trial. And we thought that when FDA approved the product, then we are done and everything is fine. But we learned that society acceptance of that drug is very critical. So I urge you today that you have to fight misinformation in a way that you have to go out and talk to the public. Learn first to talk to about your science to your grandparents. You know, if they understand, probably you can go out and talk about what you are doing and how important it is for them, so that it won't happen here, but happen in other corners of the world. Because if we are not doing, there will be always people who learn genetics or immunology in the internet, and they will fill that gap. And they will say information, which, you know, has already an effect called Dunning-Kruger effect, means that those who know less are more confident and harsher and louder. So, but of course, today I want to talk about the RNA. And messenger RNA was discovered in 1961, and it took like 60 years. Oh, no water in it. And it took 60 years.
Yes.
Okay. I just asked for the glass. Oh.
I'll fill it up.
Yeah, thank you. And that's enough now. It's not a palinka, it was water. Anyway, so it was 60 years, and every 20 years something happened. So it's other science is similar. Cas9 was discovered, people had some ideas and progress, made companies, and eventually run clinical trial. So in the case of messenger RNA, it was 2021 when in the form of COVID vaccine became approved. So it started as 1961, and I was born in 55, so I was not reading Nature in 61, but this was the paper, actually. And, you know, I have to say, so it was published 64 years ago, and believe it or not, scientists like Messelson, I just recently heard him talking, giving a lecture, or I met Gilbert, who got, by the way, Nobel Prize for sequencing, and Watson is 97 years old. So working with RNA, messenger RNA, give you long life, and you can earn a couple of Nobel Prizes because Gilbert, Brenner, Sidney Brenner, Watson get Nobel Prize. So, but the important thing is not that, but with the discovery of RNA, messenger RNA, we learn, you know, that's the central dogma of life. I know that you are geneticists, you are mostly engaged with DNA, but mRNA is just as important because it carries the information to the protein synthesis factory. So when something is discovered, like messenger RNA, scientists start to see that how does it look, and discovering long poly-A tail, other scientists already discovering, for example, how to purify, because oligo-DT was introduced early on. And with this one, you know, the scientists could look at the other end of 5' end of the mRNA. And 1975, if you work on this field, you get many, many Nature papers. Yes, and so it is very important. It doesn't matter what you know, the discovery is a technology like the oligo-DT, and so it is always helping the next scientist to step on and move forward the field. And other scientists look at translation. One of them I mentioned here. they couldn't synthesize messenger RNA, so they just could isolate. And they used the reticulocyte, which is the precursor of red blood cells, and then they could purify, and then they put in a liposome and delivered primary cells, and then they demonstrated with this Western blot here that a cell which otherwise wouldn't make beta globin, they made the protein. And I mentioned that this is a 1978, and this is a Nature paper, single author research paper with this single figure. Yeah, that scientist was easier early time to get paper in Nature. Yes, I was still in school. I was studying at the University of Szeged because, this research institute was just built and opened the same time when I went there, and 73, and I imagined myself to be a scientist. I was coming from a very small town, 10,000 people, never seen a scientist, but my biology teacher said that I could be one, and I imagined myself in white coat and this laboratory working there. So as a student, I went there, and I was interested in plants, and but there were no position there, only in lipid team. But early on, this doesn't matter, plants or lipid. And he said that to me, that I have left the rest of my life to work at the bench. I should go to a field trip. At that point, I didn't know that at age 58, I still pour the agarose gel, the polyacrylamide gel, and do everything with my own hands. So he told me that I should look at and collect some water fleas and do a little experiment, because I carried some C14 in my pocket. That was early time. And so that was the first paper I got with this water fleas experiment. And so it was, when it came back, in the fall, I went back to the lab, and two fellow scientists from the biochemistry department, Ernő Duda and Eva Kondorosi, came to the lab that they need phosphatidylserine, phosphatidylethanolamine, which was, was not available for several Deutschmark we could buy, but we were behind the Iron Curtain, and it was embargo. I don't know why it is. But anyway, these phospholipids were super secretive something. And so, but Ernő figured out there is a paper which was published, How to Isolate from Cow Brain. And I didn't mention my father was butcher, so, seeing a cow brain was okay for me. And I was started with isolating, and it took a whole week, get this fraction, because Eva and Ernő wanted to wrap up DNA and viruses and deliver to cells which wouldn't have the receptor for the virus, or deliver the DNA to the cells. And this is how it started. So this was an early paper when we delivered the DNA with liposome. And I also learned, from Ernő that, if you want to do something, you find a way. If not, you find excuses. So, okay, I learned this for the rest of my life. And then one day, the head of the RNA lab, Jenny Thomas, walked in, and he asked me that whether I want to make my PhD with RNA. And again, because other people didn't offer anything, I said yes. And so you can see that I was not a visionary person who knows that we wrap it off things lipid, and now comes the RNA. And he was an organic chemist, so he said that, oh, this molecule was discovered. This is an antiviral molecule. We need one. Even today, we need one, but it was 78. And so you should synthesize this one and set up a RNA virologic screening lab, because he was organic chemist, so it was easy for him to say. And then he left for sabbatical. And then I worked with the fellow graduate student, János Ludwig, and I was nice to him. We published together because the boss was not there. Anyway, and even today I ask help. So if graduate students are here, be nice to the fellow graduate students, so we'll help you later on. So what is this 2,5-A molecule is all about? You naturally synthesize this with oligoadenylate synthase, and then this molecule, which is unusual because this is 2', 5' linked, it will bind to a latent RNase, and it went dimerized, and that end of it cuts up, that was the idea, the viral RNA. I have to say, you know, when this was early on, I was followed rest of my life, this field. I just cannot let it go. And I learned recently that those children who ended up in ICU and COVID infected, and you know, infected with SARS-CoV-2, and their RNase L and OAS gene promoter polymorphism resulted that they had lower level of this enzyme. So it is really very important. Yes, so this was happened, and you know, we tried to you know, do this for the industry, but that I was 30 years old in '85, and on my birthday I learned that I was terminated in my position, and so I had to go to America. I never wanted to go to America. I even say that statement in America, but I had to go there, and a beautiful new building, and it was a windowless building there, and work with Professor Suhadolnik. The advantage was that I could listen great lectures, but you know, budget was low, no lab coat and other things. But anyway, and if you read my book, you learn that in America everything is bigger, because when I cleaned, you know, the cockroaches in Europe, at least in Hungary, is tiny one, but in America even that one is bigger. Yeah, so yeah, so this is what happened, and we try to develop also this 2', 5', and actually yeah, saying that cordycepin, you know, it is easy to make 2', 5' because it's 3' deoxy. And so we did that. Molecule was not, we couldn't make it to be a medicine for a human being. And again, we needed antiviral compound because at that time was the HIV. We are still 1986. So the HIV infection was very serious. And we knew that double-stranded RNA can induce interferon, but it is very toxic, therapeutic windows too small, and then we made mismatched double-stranded RNA. Later, you know, after 2001, we learned that it can activate TOL3, but at that time we didn't know. And we treated 10 patients with a high dose of this double-stranded RNA. And as you could hear, you know, I was talking here about, you know, short RNA, I synthesized long RNA and double-stranded RNA, but, you know, I wanted to talk about mRNA. I'm getting slowly to the topic. Anyway, so 1980s we are, and then they discovered how to make messenger RNA in a tube. And these were Harvard scientists, and they discovered that if it is a phage RNA polymerase, they use high fidelity, they need to provide a template, the promoter is very short, and just the building blocks are there, and then they can make human beta interferon, and that they inject it to the frog oocyte. Actually, if you had, you know, the forever young scientist like I am, so and you went to a genetic lab, they always had a terrarium with the frog. Yeah, because we didn't have the tissue culture cells. It was, you know, these are large cells, and we could inject. And they injected with mRNA, this frog oocyte incubated, and the human interferon was functional. So this gave a big push to the field, and I might mention that the scientists at the 80s were so try to help each other. I sent a letter actually to Paul Krieg, and I asked him to send me this plasmid, and he sent me, you know, in a Wattman paper, and he even put there the T7, the plasmid which had this T7 promoter to help me, because he said we made a better one. So yeah, so the hopefully intellectual property officers are not here, because this was the time at that point. And you know, this gave a big push of the mRNA research. So it could have happened that time in the 90s, because every year we get a publication. Now you are envy because it was easy to follow the literature, you know, one paper per year. But you know, these scientists, you know, like Floyd Bloom was the editor of Science, and he showed that injecting vasopressin mRNA to the brain of the rat, he could treat them from the disease. And 93, 94, they already did vaccines, mRNA-based vaccines for influenza. And the first cancer vaccine paper came out 95. And then I followed these scientists, and what I discovered that they didn't publish more papers. And I assumed that, the reason is because very small amount of protein was made very short period of time. But now that I became more known, I asked, and it turned out that they didn't get funding, and that's why they stopped. So I didn't know that I am the only one doing this and not getting funding. Anyway, I don't mention that how I ended up in Professor Suhadolnyik's lab, that I was terminated there and subject for deportation, but I fighted that and I stayed in America and ended up in the Ivy League school. That's what, that was not the last time I was kicked out somewhere, but don't expect that many termination will end up with some reward at the end. But, I was happy. Elliot was interested in this urokinase receptor. And this was because he was cardiologist, and this is a highly modified post-translationally glycosylated GPI-linked protein, and all of this decoration, all of this post-translational modification needed to have function. And we were surprised at how smart the cells were, because we just put the mRNA in it, and the cell decorated and did all of these things. And so at that point, we thought that, okay, for cell therapy, maybe messenger RNA will be good. And we were thinking like cells for bone marrow transplantation and delivering like telomerase mRNA, and the guide for that. I spent the whole Christmas time to thinking and doing this kind of thing. But when I went there, actually, I was faculty at Penn. Research assistant professor just for foreigners need a green card, but, no tenured or something. But, I was even demoted from this position after 5 years. But I stayed, and Elliot paid for 7 years my salary. And luckily, I met in Elliot's lab a medical student, David Langer. And if you have medical students in your lab, they are always the smartest one, or at least they say so. And then, that by that time, when I was needed a job, because I was, Elliot left, so he was a medical resident in neurosurgery and convinced the chairman neurosurgery needs molecular biologists. Even today is a laughable matter, but in '97, he was so convincing. So this is the place, 17 years I worked by myself. And, we did try to do different things. That invention can come, that a lot of people look at one project, every angle, or two of us. He is a neurosurgeon, and he told me that he can reach special vessels, and he told me about subarachnoid hemorrhage. I am a biologist, not a physician, so, okay, I told him that I can make iNOS mRNA, and then he can deliver there, and locally it will release the nitric oxide and dilate the vessel. So that we started to work. Budget is even lower. You can see no lab coat, no protective gears. But David left, and I met Drew Weissman at the Xerox machine, because we are in '97. Digital papers is not available, so if it would be available, I would never bump to him. Anyway, so he wanted to do a vaccine, and I want to make mRNA coding for therapeutic protein. So we started to work together, and so I made the mRNA, what he needed, and then we tested out. He could make human dendritic cells. I have to say that at that point when we met, I didn't know human dendritic cells, but it was very critical to have these type of cells. So when we treated and added with lipofectin coded messenger RNA to these cells, what turned out that they made TNF-alpha, so it was inflammatory. And we didn't understand that all mRNA is present inside the cell, and why it is inflammatory, maybe because it's coming from outside, and to test out whether all of the RNA is inflammatory. So we did an experiment At that point, I already worked like 10 years on messenger RNA and made the in vitro transcribed RNA. This is the purple here. And then I collected, I checked out all of these other RNA I collected already earlier. And what was surprising is that, these were less inflammatory, and the tRNA was not inflammatory at all. And the idea came that, there are nucleoside modification in the tRNA. I mean, 25% of the nucleoside are modified. And so this could be potential. Yeah. So Drew told me, just, the make with modification. I said, yeah, okay. I checked it out. There were 108 different modifications known in the mRNA, and I didn't know which one and how to make it. And so I started to learn. And, naturally, we have enzymes which incorporate this modification in our RNA because we make from the 4 basic nucleotides, but those enzymes is not known. And I knew 2 enzymes, actually. That was 2'-methylation and pseudoylation of the mRNA, of the RNA discovered on the ribosomal RNA, a fellow student in Hungary, that we lived in the same dormitory and we worked later in the same research center. So again, be kind to the same student in the dormitory, because you never know, you have to call him up. And I told Tamás, send me these enzymes, and because I need to modify the mRNA, but at that point was not possible. Actually, he discovered what is the function of SNO RNA. That was the guide RNA where we get the ribosomal modification, these 2 types of modification. So now that I realized that it will be difficult to make modified RNA, nucleoside modified RNA, so maybe I have to purchase some nucleotide triphosphate where the nucleoside is already modified. So I asked János Ludwig, my fellow student, where we can buy these things. And so there was 10 was available at that point in triphosphate, and I tried to incorporate and cross my fingers that the phage polymerase will incorporate, and 5 of them were incorporated. And so we try to figure out that whether it is less inflammatory. Oops. And so at that point, Drew isolated dendritic cells, primary human dendritic cells from the blood, and we tested out these RNA, the 5 of them, which I could make. And we could see that some of them were still inflammatory, but some of them was not. So when we made a closer look, we found that every time the uridine is modified, then the RNA is not inflammatory. So who knew that uridine can activate, and induce TNF-alpha? Yeah, we learned because we just opened the Nature or Science. It was Science. We opened, and that they describe that poly-U can activate TOR7. That's, happened to you also, that you just discover something. But we discovered also that how to modify. So if we modify, then it will be not inflammatory. And so that, because I want to make mRNA, which is, code for the therapeutic protein, Drew wanted vaccine, so it was important that it is translatable. So this is what we did, and that was the surprise that pseudouridine-containing RNA, we get 10 times more protein, whether it was primary cell or 293 cells and others, and some others was not translated at all. So that was a good news that we can make RNA which is non-inflammatory and which translates so well. So it was like the dream comes true. And what happened in the United States, you usually file a patent. It's not easy because it costs money, and I might mention here that when you go to the patent, the IP office, they ask you whether it is useful for something, because nobody will go to the university IP office and say, "Oh, pseudouridine, oh, I will invest that." You have to form a company and do something. But anyway, so we went to the office, and we will ask what is useful for this pseudouridine RNA, and because he happened to be not to have a hair, he was a bald guy. I said probably hair growth. Yeah. So this is up to now scientists that prove that, because it could be. you just have to try. But anyway, we did a company and formed a company, RNARX, that you never heard, because we never get our own patent for our own company. But we wrote a grant, small business grant, and that's the only grant I ever get, is $1 million from the American taxpayers' money, and it paid my salary like 4 or 5 years. And so we promised in this grant that we will make erythropoietin-coding mRNA, which potentially can treat anemia. And we did in animal studies, we complex with transit this messenger RNA code for erythropoietin, which can generate red blood cells. And then when we injected, I mean, you are working with genomic material, so 0.1 microgram. So you know that it's quite a small amount, injected to the animal, and when it was pseudouridine containing, that even 4 days later, we could see the protein was still present in the circulation, and the half-life of EPO protein is just 2 hours. And it was functional because, you know, the hematocrit increased, and the weekly injection of the animals resulted that the hematocrit, high hematocrit could be maintained. So those mice were very happy and jumping in the cage. And luckily, you know, that there were no cytokines was induced by that. And so it, at that point, seemed that everything is going well. We are 2012. I was happy that my daughter got the second Olympic gold medal, yeah, in London, and then, yeah. So usually, you know, that people, when they say that, Kati, you know, you were not known, I said, oh, American Rower knew that I was Susan's mom. And so, but in these days when she comes with me, usually she is Kati's daughter. But yeah, so things changed. But anyway, in that time I was terminated at University of Pennsylvania. I forced to retire because I didn't bring in the money. And so I have to say, you know, it could happen to you. And it is very important that don't start to feel sorry for yourself. Go to your colleagues, say, you know, it is not fair. I was here all weekend. I was working so hard. And don't waste your time with that. You know, after 24 years, I walked towards, you know, my car and the parking lot. Nobody gave me a goodbye party, nothing. I was just leaving and was focusing on what next. And that's what I recommend. Don't ever, somebody already made a decision, but don't let other people define you. So you have to believe in yourself that, okay, you can do some more. And I was 58 years old. I felt I was the top of the game because finally I get 2 job offers. You know, I loudly said, old lady still be wanted. And I get one in Moderna and one in BioNTech, vice president. You know, when the company is very small, you walk in, you might get high position. Anyway, I ended up in Germany because it was far away from home. And I worked with Ugur and Özlem. And we were not in this fancy building, we were in the campus, but we further improved the RNA. And this was, you know, now that is more protein was produced and longer time because we codon optimized, we purified and did many, many things. And you know, more things led to this, and I made a cartoon for here for you, just to understand that, you know, to make an mRNA, it was important where we will get the DNA, and that at the beginning I had to beg to fellow scientists who cloned certain genes to send me that. And then it came, you know, in the 80s, the RT-PCR, TA cloning, probably you have done that one. And then later we could order genes like from Origin or somewhere, which is already a fixed gene. And from 2000, like you could order a gene, whatever you wanted to incorporate in. I put in 2006 because at the beginning it was $4 per nucleotide. I didn't order. 2006 I checked, and that was the first time I ordered. And so, you know, that was made easy. And you know that even if the COVID would, you know, this pandemic would come 20 years earlier, you know, information would not be enough. You would need a little package from Wuhan, no how much you don't want to get one, but to start to make a vaccine, you needed some genetic material to start. And the field improved a lot with translation when CAP was discovered, like Pharmacia already made CAP analogs, others start to sell polymerase, capping enzyme. You could also buy CleanCap, which you could incorporate transcription. You could modify the RNA. And of course, more we get from delivery, because you had to protect that RNA and deliver, and then it released. And eventually, we get a lipid nanoparticle. And I made this just for, I couldn't stop to think about, I went to America in '85. Can you imagine there were no gene bank, no PubMed? Today you think that it was forever there, and for me it was listening great talks, but today, 20 years ago was YouTube, so you can listen to anybody, so many, and the problem is now that who you should listen, because so many choose from. But everything contributed to the success by the vaccine could be developed so quickly. Even I can say Twitter, because Yang-Sen, who sequenced in Shanghai the SARS-CoV-2, he told me that he put to the gene bank, but people did not pay attention, and he asked his friend at Holmes to take out to the Twitter to tell the people that the sequence is there. So, everything, the sequencing that in Shanghai, everywhere on the world, you can sequence. The companies can, somebody had to discover how to do it, improving, and so everybody contributed to the success. And why I am talking, why it is important, the mRNA is more and more protein is available as a, FDA-approved product, but they are very expensive. So if we can replace the protein-based drugs with mRNA, so let the patient to make their own drug. You don't need purification, proper folding, and so on. And when you read in these days, there are so many different RNA. It can be modified or unmodified. It can be self-amplifying. These are already approved, self-amplifying already in Japan as a booster. And there are, you can read about the new one, the circular RNA, which internal ribosome entry site or CAP, covalently or not. And the circular RNA, is the novelty they don't know about. In '94, I already had a grant which was rejected on circular RNA. But yeah, in these days, I have to say that 150, at present 150, more than 150, messenger RNA is in human clinical trial. Yeah? And half of them is vaccines. Most of them is viral vaccine. I listed here, which are in clinical trial, like Nipah is phase 3, and monkeypox, others. There are bacterial one, like tuberculosis. Borrelia is for Lyme disease, or parasite like malaria. Human trial is ongoing. And for cancer, you can hear more about that. There are some, shared tumor antigen targeting. Others is individualized, so only the one person who had that tumor and neoantigen can get that. And there can be used, I was working on messenger RNA coding for bispecific antibodies, so that the patient can make the very expensive antibodies, and mRNA is cheap. And they can produce it. And at the beginning, actually, Moderna published or reported before 2020, the cardiovascular disease use of VEGF mRNA, which generate new blood vessels, and they treated patient heart failure, injected directly naked RNA to the heart, and they also did treated wound, necrotic wound in a diabetic patient that didn't heal well. And the genetic disease, you are talking here about these different treatment for there are several of them which directly delivering mRNA, which is missing enzyme, mostly in the liver. And for cystic fibrosis, several trial was run already and presently is ongoing. And some of them is, gene therapy. So once 5-10 years ago, we wrote with Drew Weissman that maybe the promise of gene therapy will be fulfilled by mRNA. Because, if you deliver the Cas9 mRNA, and then you can change the genome. And there are more, you can read about autoimmune diseases. They made CAR T cells with mRNA, and for myasthenia gravis, it is already phase 3 trial, and for lupus is phase 1. And not just the human, animals, even elephant in Texas benefiting from mRNA vaccines, and horses were injected. I don't know, the shrimp maybe is a little challenging, but they did the duck actually in France, and it was a replicative RNA. What left is for me is just inspire the next generation of scientists. Here with Drew Weissman, you can recognize us. And this was written by others and with different languages. But I myself wrote a book about my life, and it is published in many different languages, from Vietnam to China and others, and is coming out in Iran and Serbia soon. And so, I never wanted to write a book, just people approach me that they want to write about me, and then I said, who cares about the boring life of a scientist? Because what we are doing outside is like boring, pipetting at least. So, but, eventually, they convinced me, and then I told my daughter that you should write, she's good. Anyway, so this book were published, and in Italy, actually, 2 weeks ago, I got an award for my book from high school student, 14,800 voted, and because this was professors and students from the universities suggested books for the high school students should read. And there were 5 books were selected, and 2 books, one of them was mine, which was selected by the students that they can learn from it. So we need to have the next generation, the kids who are in elementary school or high school. And so when I get the award, and today I get, but I have to say, thank you for that. And usually, I say thank you, to my families, all of the teachers, and who I thank that I am here. I usually mentioned all of those people who made my life difficult. I am also thankful to them because they made me work harder and more resilient. And when I get the Nobel Prize, okay, I didn't invite those, only invited those who were my family members, and my daughter, my husband. And without my husband, I wouldn't be here today. And my teachers for biochemistry, genetics, and here is the Erno and Eva. We did the first liposome experiment. And of course, David, the famous medical student who now is chairman of neurosurgery in New York, and Elliot, who hired me, they were all my guests. And I am very grateful for all of the people. And please, I ask you also, from time to time, say thanks to your mentors, to your teachers, to your family members, because, scientists, okay, we don't have this nice work-life balance now. We scientists immediately we forget about, and we still have, our partners and our children, and they suffer a little bit. But I believe that, showing the good example, what we are showing, that it counts more. I try to explain always, to make me less guilty, that it is good for her to see me work hard, and then she just followed. And with that, I thank you very much for your attention, and I wish you enjoy the conference.